Hello everyone. Today we are going to talk about membrane potentials, especially RMP, what exactly it is and how it is formed. So RMP, it stands for resting membrane potential, and it is defined as voltage difference or potential difference across the cell membrane when the cell is at rest or in the non-excited state. It is basically the difference between the electrical potential in the cytoplasm of the cell and the electrical potential in the extracellular space. You can see there's excess of positive charged ions in the extracellular side, excess of negatively charged ions in the cytoplasmic side. So the point which I want to convey is due to this separation of positive and negative charged ions, this results into membrane potential. Now, in order to have potential difference across a membrane lipid bilayer, two conditions should be met. First one is there should be concentration gradient. What do I mean? There should be unequal distribution of ions of one or more type across the membrane. And secondly, the membrane it must be permeable to these ions. This permeability is provided by channels or pores in the bilayer, and these channels they are usually permeable to single type of ions. Next is RMP is present in almost all cells of our body. Every cell of our body has certain membrane potential across its membrane. But if you talk about action potential, only special cells, that is, neurons and muscle cells, have this ability to generate action potential, be it smooth muscle, be it cardiac muscle, be it skeletal muscle. Now, the question arises why only neurons and muscles? It's because of their excitability property. They are excitable cells and they can have a transition from resting state to an excited state. Whenever they are stimulated properly, they can generate rapidly changing electrochemical impulses at their membrane. And these impulses they are used to transmit signals along the nerve-muscle membranes. So action potential is nothing but those fluctuating electrochemical changes across the cell membrane which is produced only in the neurons and only in the muscle cells. Now, let's just take a hypothetical cell with ECF around, that is extracellular fluid, the fluid which is outside the cell. See, before that, I would like to mention, as we all know, life started in ocean. Our first life started in ocean according to evolution theory, and ocean water is salty. So, what I am trying to convey is our mechanisms are evolved with salty water around. Every cell up to now in human body, it is keeping the salty water around it. This is that salty water. This is that internal sea which every cell is keeping around it, and this fluid is called as extracellular fluid. If you take this ECF out and compare it with the present seawater, you are going to find this more dilute. But if you look at its composition, then it closely resembles to that of the primitive oceans where life, possibly originated. So that was possibly the logic why cells have high concentration of sodium in ECF. Here is a brief overview comparatively. If we talk about inside the cell, intracellular, then there is high concentration of potassium and negatively charged anions, which includes amino acids and proteins produced by the cell. But if you look outside, that is in extracellular fluid, there is high concentration of sodium, chloride, and calcium plus 2. Now, let's just take a hypothetical cell over here. And of course, the cell is nucleated. Inside nucleus, we have genetic material. And as I told you earlier, every cell it's trying its best to keep more and more sodium outside. So for this purpose, we have this special kind of gene over here. And this gene is present in every cell. Whenever this gene expresses, it is making a protein that gets implanted into the membrane. And this protein is a special kind of sodium-potassium transporter. This is known as sodium-potassium pump. This is also known as sodium-potassium ATPase. I'll explain you later in the next slide why it is known as sodium-potassium ATPase. So what this protein does is, it keeps on throwing sodium outside the cell and it keeps on concentrating potassium into the cell. This protein, it keeps on pumping sodium outside despite of the fact that there is already a lot of sodium outside. So it is pumping sodium against its concentration gradient because concentration of sodium is outside more and it is still pumping sodium out. On the other hand, it is constantly bringing K plus into the cell despite of the fact that cells are already rich in K plus. Still it is pumping more K plus inside. So it is bringing potassium into the cell against its concentration gradient. So this protein, it is doing an uphill task, that is active transport. To be specific, it is primary active transport. In the next slide, we have this pictorial representation of what a sodium-potassium pump embedded into the cell membrane looks like. And here is a simple representation of what I have drawn for your understanding. As I told you, cells are rich in K+, so they are bags of K+, floating into the sea. And that sea is what? Extracellular fluid, which is rich in sodium. This sodium potassium pump, it pumps 3 Na plus out of the cell and 2 K plus into the cell for every 1 ATP consumed. This carrier protein has 3 binding sites for sodium on the portion of the protein that protrudes on the inside of the cell. This is the portion of the protein that is protruding inside, and here we have 3 binding sites for sodium. 2 binding site for potassium on the outside. This is the portion of the protein which is protruding outside, and here we have 2 binding site for potassium. Next we have is on the inside portion of the protein near the sodium binding sites we have ATPase activity. In the diagram, as you can see, on the inside portion of the protein near the sodium binding sites we have ATPase activity. ATPase is an enzyme which stands for adenosine triphosphatase, and this enzyme, it catalyzes the hydrolysis of phosphate bond in ATP to form ADP and a free phosphatine. See, this enzyme, it basically breaks the high energy phosphate bond between second and third phosphate group in ATP, and this hydrolysis reaction, it releases energy. See, that liberated energy, it causes a chemical and a conformational change in protein, resulting in 3 Na plus out and 2 K plus N. That is the reason sodium potassium pump is also known as sodium potassium ATPase, because it has ATPase activity on the inside near the sodium binding sites. See, this sodium potassium pump, it's very crucial because it is found in plasma membrane of almost every human cell. And this pump, it is literally the basis of nerve function because it helps in transmitting nerve signals throughout the nervous system. See, let me give you an estimate. Near about 20 to 40% of brain energy consumption is just due to activity of this pump. Not just this, in fact, in some electrically active nerve cells, 60 to 70% of energy is just devoted to this pump. So from this you can get an idea how important this is. Now let's suppose this protein, it works 100 times, 100 cycles. So basically in one cycle the stoichiometry was 3 is to 2, 3 Na plus out and 2 K plus N. So in 100 cycles it is going to be 300 Na plus out and 200 K plus N. So basically Na plus and K plus both are cations. So what is happening is 300 cations out and 200 cations in. So there is net loss of cations. So the total amount of cations lost from the cell are more, but the total number of cations gained into the cell, they are relatively less. So practically the cells are losing cations outwards. So it means cells are gradually getting electronegative, because we are losing 300 cations outwards but only gaining 200 back in. So there is loss of 100. So eventually the cells, they are going to get electronegative. See, next we have is sodium potassium pump is an electrogenic pump which is making cell interior slightly negative by minus 5 millivolt as compared to exterior. Now what does an electrogenic pump means? Any pump which is working across the membrane, and during its function if it is creating an electrical imbalance, then that is called as electrogenic pump. Now what imbalance is sodium potassium pump creating? So basically sodium potassium pump is pumping 3 Na plus out for every 2 K plus N, and due to that it is leaving a net deficit of positive ions on the inside, and that is causing a negative potential to develop inside the cell membrane. As I told you, by function of sodium potassium ATPase, interior of cell, it becomes electronegative as compared to exterior, but only by minus 5 millivolt. Now what does this minus 5 millivolt indicates? See, whatever sign is in front of this number, this indicates the interior situation of the cell. As it is negative here, it is indicating that inside of the membrane is negative. And whatever numeric value is here, it is the potential difference between the inside of the membrane versus outside. As I told you earlier, membrane potential is the potential difference across the cell membrane, that is between the inside and the outside. So basically, the membrane potential is present on the inner side of the membrane compared to the outside. So the outside is used as a reference and we assume it as 0. Alright, so whatever number we talk about are on the inside. Let's suppose if we say plus 60 millivolt, Then plus this is indicating that inside of the membrane is positive, and this numeric value is the potential difference between the inside versus outside. Now, if you look at the values of RMP, it has a range of minus 70 millivolt to minus 90 millivolt. See, it varies from cell to cell. Most of the neurons they have this RMP of minus 70 millivolt. But if you look at cardiac muscle cells and skeletal muscle cells, then they have this RMP up to minus 85 millivolt and minus 90 millivolt. So basically, inside of the cell membrane, they are electrically negative, approx minus 90 millivolt as compared to outside. But if you look at the contribution of sodium potassium pump to this RMP, then this is very little, only minus 5 millivolt, a little contribution to RMP. Let's just say if we start from 0 millivolt, then this sodium potassium pump, it helps us to reach minus 5 millivolt. Still, there is a long way to go to reach minus 70 millivolt. In the next video, that is part 2 of RMP, we are going to study about leaky channels, diffusion potential, Nernst equation and all of that. Thank you so much.